My name is Parada Peerachidacho. The subject of my talk is population genetic structure of Tylototriton panhai from Phuket National Park, Phitsanulok Province, Thailand. Okay. In the first part, I will introduce about genetic diversity. Genetic diversity is the variation of alleles and genotype among individuals in a population. Genetic diversity controls morphological, physical, and behavioral variation and cause population to adapt to environmental changes. So, loss of genetic diversity may reduce fitness of populations, followed by population decline, and finally lead to species extinction. Therefore, genetic diversity is one of the major components commonly used for population assessment and conservation of biodiversity. Habitat loss has long been considered as a major factor in a population decline and extinction to reduce genetic diversity. Amphibians are top concern of population loss because they inhabit both terrestrial and aquatic environments. Moreover, amphibians have low dispersal ability, so they are unlikely to move into new habitats. Amphibians play important roles in ecosystems. Their diet consists mainly insects, so they play an important role in controlling insect population, particularly disease vector and agricultural pests. Unfortunately, amphibian populations are decreasing worldwide, while there are few studies about genetic diversity and conservation. Tylototriton is the only genus of order Chordata currently recorded in Thailand. In Thailand, they are distributed on the high mountain with the elevation higher than 1,000 meters above mean sea level, as shown in this map. To date, 5 species was reported in Thailand: Tylototriton uenoi, Tylototriton verrucosus, Tylototriton anguliceps, Tylototriton phucaensis, and Tylototriton panhai. For the conservation status, according to IUCN, Tylototriton verrucosus and Tylototriton anguliceps are categorized as least concern species or LC, while the other Tylototriton are categorized as data deficiency or DD. Moreover, only Tylototriton verrucosus is the amphibian wildlife protection list of Thailand, or บัญชีสัตว์คุ้มครองประเภทสัตว์สะเทินน้ำสะเทินบกของประเทศไทย Although most of news reside in the protected areas such as national park and wildlife sanctuary, they are threatened by extinction mainly by human activities such as destruction and pollution of their habitats, deforestation, and so on. In my study, I focus on panhai crocodile or Tylototriton panhai that are distributed throughout the Phetchabun ranges, which are Phuket National Park, Phitsanulok Province, Phu Soi Dao National Park, Uttaradit Province, Phu Suan Sai National Park, and Phu Luang Wildlife Sanctuary, Loi Province. Phuket National Park is the southernmost distribution range of this species. Moreover, Phuket National Park is a famous tourist destination where a large number of tourists visit the park each year. There are 2 major breeding ponds of new have been reported at Phuket National Park, Headquarter and Suburb. Because of Tylototriton panhai is considered as a sensitive species based on its patchy distribution and isolated population, so different genetic structure between these 2 breeding areas may exist. Thus, this study aims to examine population genetic structure and genetic diversity of Tylototriton panhai from Phuket National Park. The result will be used for future management and conservation purposes. The field survey was conducted in May 2019, which was the breeding season of Tylototriton panhai. News were collected from their breeding pond at headquarters in Tabuk during the raining season. The tail were removed and preserved in 95% ethanol for future molecular analysis. Then the wound was cleaned with an antiseptic liquid. Total DNA was extracted using pure genomic isolation kit. The mitochondrial DNA DH3 hydrogenase subunit 2 or 82 gene was amplified by polymerase chain reaction or PCR using specific primers and conditions. Then the PCR products were checked by gel electrophoresis technique to confirm size and concentration. PCR products were purified and sequenced. Next, all sequences were manually checked and trimmed. Then the alignments were conducted genetic diversity indices, including number of haplotypes, haplotype diversity, nucleotide diversity, genetic differentiation, and pairwise genetic distance. The median joining haplotype network was constructed to examine the relationship of Tylototriton panhai population at Phuket Long National Park. The PCR products were pure. Oh, sorry. Okay. ND2 gene sequence review 1035 base pair. Number of haplotype diversity, and nucleotide diversity of headquarter population were relatively higher than those of Tabuk population. Since the Tabuk is a tourist destination and easily accessible by cars, human activity may cause negative impacts on the new habitat, including ecosystem and resource degradation. Moreover, the surrounding area of Tabuk may receive irreparable damage from tourists and subsequent population decline. The result decrease in the effective population size could result in decreased genetic diversity in a population. On the other hand, the effective population size may be affected by breeding pond size, as previously reported in California tiger salamander or Ambystoma californiensis, which show positive correlation between effective population size and breeding pond area. In this study, the breeding pond at Tabuk was a small permanent pond, while the breeding pond at headquarters was a part of a long, slow-moving stream and was a comparatively larger in size. Thus, the pond at headquarters may serve as more suitable habitat supporting larger population size. So, Tabuk population size was possibly smaller compared to headquarters population and result in relatively lower observed genetic diversity. However, unequal sample size in this study may have affected the genetic diversity indices as well. The number of haplotype diversity, and nucleotide diversity obtained from this study were rather similar to those previous reports in tiger salamander or Ambystoma trigonum, which was also categorized as least concern species. However, the tiger salamander was comprised a large number of subpopulations and locations, large number of population size, and have no signal of anthropogenic population decline. Thus, those factors must be considered to determine the conservation status of Tylototriton panhai. The result of this study revealed relatively high haplotype diversity, while the nucleotide diversity was relatively low. The results suggest that the small population were undergoing rapid population growth, the time period of which was sufficient for genetic differentiation, but which present of high haplotype diversity, but not accumulation of nucleotide difference, which present of low nucleotide diversity. The result of genetic differentiation and pairwise genetic distance show significant genetic difference between headquarter and topbird populations. Moreover, the median joining haplotype network reveal 8 haplotypes, including 7 unique haplotypes, which are pH 1, 2, 3, 4, 5, 7, and 8, and only one shared haplotype, which is pH 6. Some Tylototriton species are philopathic or trending to return or remain near their breeding point. This behavior may restrict inflow between populations. For this reason, Tylototriton panhai may also be philopathic to their breeding point, and restrict inflow may have resulted in observed genetic structure between headquarter and topbird. Furthermore, these two populations may have shared the same gene pool in the past, was evident in the only one shared haplotype in this study. From this study, headquarter new population reveal relatively higher genetic variation compared to topbird population. So anthropogenic effects, which may have resulted in population decline and loss of genetic variation, should be evaluated. Although the genetic diversity indices of Tylototriton panhai at Phu Long Kra National Park were similar to other salamander species categorized as least concern species, there is still lack of long-term population monitoring data in Tylototriton panhai, which is necessary for determination of conservation status. There is significant genetic differentiation between headquarter and topbird population. This suggests that each population should be considered genetically unique, and both populations are important for maintenance for the overall genetic diversity. This is particularly essential to ensure physiological adaptation to environmental change, survival of species, and preservation of biodiversity at Phu Long Kra National Park. We would like to thank the following. Thank you for your kind attention. Any questions?
Yeah. Thank you very much for your interesting presentation. Okay. Are there any questions from the audience? Okay, I have one question. Okay, that your presentation, okay. You mean that the accumulation of nucleotide difference you found only in population of topbird is correct? The accumulation of
nucleotide difference, right?
The accumulation of nucleotide difference was observed in topbird population. It is correct?
No, actually, it's in both populations.
In both populations. Oh.
Yes.
And what?
Yeah.
What's the definition of accumulation of nucleotide difference? I want to know this.
Okay, I will explain you about this. Right, this you mean it?
Yes.
Okay. From this result, I found a high haplotype diversity and low nucleotide diversity. The definition of haplotype is the pattern of the gene. Okay, I represent the one pattern of gene or one haplotype is a color here, is a circle color here. So if you imagine from the small population and they have a rapid population growth, become a large population, and the pattern of gene is there are many patterns of gene, so it's present in the high haplotype diversity. But if you look deep up in the haplotype and you found the sequences, and you found that the nucleotide in the sequence is still have a little number of the variation size in the sequence, that means it's still low nucleotide diversity. If you understand it.
Yes.
Okay, thank you.
Do you have any question? I think it's like, I think this topic is quite interesting. Okay.
Thank you.
Do you have any question from the audience? If no any question, please thank you, Miss Parada, very much. Thank you very much.